So in this video, we're gonna start with chapter number 14 and we're gonna talk about or start a concept of the biomolecule. There are 4 biomolecules that we need to study in this course. And what are those? One of them is carbohydrate. Then later on, we're gonna talk about the proteins. Then we're gonna talk about the lipids. And the last, we're gonna talk about the nucleic acid. So these are the 4 biomolecules that we're gonna start and talk about later in the course. But today our focus will be on one of the biomolecule, which is a carbohydrate. And in this video, we're gonna talk about what are the structure of several carbohydrate, what are the different types of carbohydrate, and what are their function. So let's get started. So this chapter is normally divided into different subtopics. First, we're gonna talk about the overview of the carbohydrate, what is carbohydrate, and what are their functions. Then we're gonna talk about one of the topic called stereoisomers, and after that we're gonna talk about the structure of the common monosaccharide. What are the different monosaccharides we have, and what are their structure. Then later on we're gonna talk about the structure of the complex carbohydrate, and at the last we're gonna talk about the role of one of the carbohydrate called oligosaccharide, and see what's the role of these carbohydrate as a cell marker. So let's get started with our first topic, which is an overview of the carbohydrate and their function. So now let's talk about the first subtopic, which is an overview of the carbohydrate and then their function. So I'm guessing you might probably already know about the term what is carbohydrate. So carbohydrate is a biomolecule that is very important to sustain the life on Earth. And it's been found that half of the carbon on Earth is found in the form of a carbohydrate. So whatever the amount of carbon that we have on Earth, they are present in the form of a carbohydrate. But how would the carbohydrate produce on Earth? So carbohydrate is normally produced on Earth through a process called a photosynthesis. And who does this photosynthesis? Plant does the photosynthesis with the help of a sunlight and carbon dioxide and water, and they produce a carbohydrate and oxygen as a product, right? So carbohydrate we get to get an energy, and oxygen as a byproduct we normally use for breathing. So the reaction will normally go like this. So carbon dioxide react with water in the form of a sunlight, right? What is this sunlight? This is an energy source, and when plant get all these reactant, it produce a carbohydrate C6H12O6 and oxygen as a byproduct. So this is your carbohydrate, and this is a byproduct of this reaction, which we know you normally use for breathing, oxygen, right? So once we got a carbohydrate, so who use carbohydrate? Plants and animals use this carbohydrate, right? And then whenever we eat carbohydrate, we get energy through a process called cellular respiration. So cellular respiration is a process where we normally use a carbohydrate as a source of energy and break down carbohydrate into a simpler compound, and then it releases energy, and that energy normally being used by the animals to do their cellular function or any activity, for example running, walking, playing, right? So how does this reaction goes? So this reaction normally goes something like this. You have carbohydrate C6H12O6, you break down into a product H2O, and then you release some energy, and this is the energy that normally being used by the animals or plants that normally use to carry out their function, right? Like I already told you, running, eating, right, walking, doing some work, right? And then if you can see over here, which is a product, one of the product called carbon dioxide that we breathe out, then it's go to the environment again, and in the environment plant take out this carbon dioxide again to do a photosynthesis. So if you can see, this is a cycle, right? First carbon dioxide react with water to produce a carbohydrate. Once we eat a carbohydrate, we break down the carbohydrate again into a carbon dioxide and water and release energy, and this same carbon dioxide goes into the environment again and goes into the photosynthesis pathway again. So this is like a cycle. So photosynthesis and cellular respiration work simultaneously, right, one after another. So there are two kind of pathway that we're going to talk about. One of them is called as anabolic pathway. Let me write it down over here, which is anabolic pathway. So what is anabolic pathway? Anabolic pathway is the formation of a larger compound, in this case the carbohydrate, with the help of energy, right? And then we have a second pathway called catabolic pathway. And what is catabolic pathway? Catabolic pathway is that pathway where we normally use a carbohydrate, any source of energy, to break down into a multiple product, and we get energy from there, right? So cellular respiration is a catabolic pathway because we are breaking down the carbohydrate into a multiple product, and we are getting the energy from there. And photosynthesis is your anabolic pathway because we are making a larger product, which is a carbohydrate, with the help of energy. So we are using energy or store energy, right, in the form of a carbohydrate. So this is an anabolic pathway. So in our body, these two pathways going side by side, anabolic and catabolic. Then let's talk about the function of the carbohydrate. What are the different functions that carbohydrate does in a cell? So carbohydrate does multiple function. Some of them are, carbohydrate is an important part in the structure of the nucleic acid. So it involves in the structure of nucleic acid, and we know what a nucleic acid like DNA or RNA. And the second important function that carbohydrate does are cellular recognition. It helps the immune system of our cells to recognize that this cell belongs to the same environment. It's not a foreign cell, right? So we're gonna talk about this, how this carbohydrate helps in a cellular recognition within the body. Okay, so now let's go to the next slide, and then let's talk about the carbon cycle. So in the last slide we talk about the photosynthesis and cellular respiration, right? And what we got in the last slide is photosynthesis is a process where plant use carbon dioxide and water with the help of energy, input of energy, right? It is do a photosynthesis reaction and to produce a carbohydrate and oxygen, right? And once the carbohydrate is eaten by the humans or any animals, they get energy, right? Because carbohydrate is going to break down into multiple product, right? Carbon dioxide and water, and it releases some energy, and that energy is being utilized for some activities like running in this picture, right? Like eating. And then that energy that is being released by breaking down of the carbohydrate in our body, it released in the form of a special chemical called ATP. And we're going to talk about what this molecule is and later when we talk about the DNA, right, and the metabolic pathway. But right now just remember ATP is like an energy, right? And then what is the breakdown product? It's carbon dioxide and water. So if you can see, then this carbon dioxide again go to the photosynthesis. So this is a cycle where carbon is being used and synthesized back again. So this cycle is we call as a carbon cycle, right? So remember what is carbon cycle, and it is a combination of photosynthesis or cellular respiration where the carbon dioxide is being used and then released, reused and released, right? So now let's talk about the different types of carbohydrate. What are the different types of carbohydrate we're gonna study in this chapter. So but before we're gonna talk about what is carbohydrate, So it's been seen previously that carbohydrate is a hydrate of carbon. That's what the name is, carbon hydrate. So carbon, CN, and hydrate. Hydrate is your water. So it's been seen that the carbohydrate is a hydrate of carbon. So this is your hydrate. This is your carbon, right? And N is the number of carbon that you have, right? N is your number of carbon. Number of carbon, right? But it's been seen that later on that carbohydrate is not a hydrate of the hydrate of water. Basically they are polyols. What is poly? Poly means many. All mean, if you remember from the organic chapter, alcohol was the ending word for alcohol, all. So this is your alcohol or hydroxy part. And what is this? OH. So it's been seen that carbohydrate is a polyol that contain either an aldehyde or a ketone functional group. So you will always see one of these functional group on carbohydrate, either an aldehyde or a ketone. And let's talk about what is aldehyde. We already talk about aldehyde is CHO. That's your aldehyde. And what's your ketone? Ketone is R C double bond O R. That's your ketone. So make sure you remember this functional group, because we're gonna use this functional group a lot in this chapter. Right. And then how we name this carbohydrate, the ending word of this carbohydrate is always end with O's, right? For example, glucose, fructose, right? And carbohydrates are normally classified by the number of monosaccharide produced when hydrolyzed in the presence of a catalyst. So we're going to talk about what are the type of carbohydrate later on, what are monosaccharide. But right now just remember, sometimes carbohydrate are also called as a saccharide, right, which is a sugar, right? So if you see the word saccharide, means we are talking about the carbohydrate, right, or a sugar most probably. So let's go to the next slide and then talk about what are the different types of carbohydrate or a saccharide we have. So let's talk about the types of carbohydrate. There are three types of carbohydrate. One of them is monosaccharide, second one is your disaccharide, and third one is oligosaccharide. And then we have fourth one on the next slide, which is polysaccharide, but we're gonna talk about that thing later on. But right now let's focus on the monosaccharide. Mono means one, saccharide means sugar, right? So monosaccharide, one sugar. So these are the simplest carbohydrate and it cannot be hydrolyzed. So if you want to hydrolyze, what is hydrolyze? To break down the bond, right, with the help of a water. So if you want to break down this monosaccharide with the help of water, you won't see any product, because this is the simplest carbohydrate, right? So monosaccharide is the one sugar, simplest carbohydrate. Let's talk about the disaccharide. Di means two, saccharide means again sugar, right? So two sugar. So whenever you do a hydrolysis reaction with disaccharide, how many sugar you gonna get as a product? Because it's made up of two sugar. Let's take an example. This is sugar number 1 bonded with sugar number 2. So when you do a hydrolysis reaction, so this bond is going to break and you're gonna make two sugar as a product, right? Plus sign, 1 2. So what is this product? These are monosaccharide, right? So whenever you do a hydrolysis reaction on a disaccharide, you always end up with the monosaccharide, two monosaccharide, because the disaccharide. Then we have oligosaccharide. Oligo means few, right? It normally goes from 2 to 5 sugar molecule. So whenever you're going to hydrolyze this oligosaccharide, you're gonna get few monosaccharide. It can be 3, it can be 4, it can be 5, but it won't be like more than 10. But yeah, so make sure you know how to define mono, di, and oligosaccharide. Let's go to the next slide. So let's take an example of the oligosaccharide. If you have this oligosaccharide which is bonded with each other, this is sugar number 1, 2, 3, 4, 5, and if you do a hydrolysis reaction in the form of water, then what's going to happen? This bond is going to break, right? And then you're gonna get 5 different monosaccharide, or 5 monosaccharide, plus 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 and then plus. So they are separate now, 3, 4, 5. So this is how you normally get a product. So now let's go to the other slide and talk about the polysaccharide, the 4th type of carbohydrate. So the 4th type of carbohydrate is a polysaccharide. Poly means many, right? Saccharide means sugar. So polysaccharide means whenever you hydrolyze a polysaccharide, you will get many monosaccharide, like 20, 30, depending on how many monosaccharide normally used to make that polysaccharide, right? So it's multiple. It can be 20, it can be 100, right? So what are polysaccharide? Polysaccharide are a biological polymers with monosaccharide as a monomer. So what is polymer? So polymers are the large molecule that is made up of multiple smaller unit called monomers. These are the subunit, right? So polymer are the large molecule that has a subunit of monomer. When multiple monomer combine, you get a polymer. So polysaccharide is a polymer of a monosaccharide. So monomer in polysaccharides are monosaccharide, right? So just remember this concept. Then we already seen this thing. Monosaccharide exists as a stereoisomer, and we're gonna learn about what this term is, stereoisomers, on the next slide. But just remember, monosaccharide can show an isomer called stereoisomer. So now let's go to the next slide and talk about this special term. Now let's talk about the second topic in this chapter, which is a stereoisomer. So stereoisomers are the compound with same molecular formula and same connectivity of the atom, but different three-dimensional orientation of the atoms, right? What does it mean? Let's say if you have any connectivity of atom A, B, C, D, E, right? So if I have to make any more structure with the same compound, something like this: A, now it's got D, it's got C, it's got B, it's got E. So what's happening between these two? They have the same molecular formula, which is A, B, C, D, E, but with the same connectivity. If you can see, every atom in both the structure connected with an A, right? Same connectivity, but different 3D orientation of the atom. But what's the difference between two? In the 3-dimensional space, then the orientation is different. Over here in the first picture, a compound, the D is present on the bottom of A, wherein in the other one is present on the top. So it's a different orientation in the 3-dimensional space. So this is we call as a stereoisomers, a special type of isomer. Then we already talk about the geometrical isomer in the previous chapter, what a geometric isomer, normally shown by the alkene, right? Because you cannot rotate this carbon-carbon double bond, so that's why we have a special kind of isomer called geometric isomer. And this is different than the stereoisomer, because you can rotate a single double bond, a molecule, right? Whereas in geometrical isomers you cannot rotate this double bond, and because of that, but you need one special requirement before any compound can show you this stereoisomer, and that requirement is the molecule or a compound has to be a chiral. If a molecule is not a chiral, then they cannot show a stereoisomer. So you need to know what is this chiral mean, right? So let's go to the next slide and then talk about what this term, what is chiral, and how would you determine whether the compound is a stereoisomer if they have any chiral molecule in there, right? So let's go to the next slide. So now let's talk about the term called chirality. Chirality is a symmetry property of some object and molecule, also known as handiness. So let's take an example and to learn about the chiral object, right? What are chiral objects? So let's take an example of our both hand, left hand and right hand. So if and then we are going to try to make an analogy with the molecule with our hands, right? So if we see our left and right hand, if you can see, we have the same numbers of fingers, right? One thumb, four finger in both of our left hand and right hand. So we can try to make an analogy with same formula in the molecule, right? Because both the hands have same formula, one thumb, four finger, another hand same, one thumb, four finger, means same formula. And then if you see the sequence, how the fingers are arranged, you will always see the thumbs on one side, and then we have four finger. Then on the other hand, you will have a thumb on one side, four finger on the other side. Means they have the same connectivity of same sequence, and we can make an analogy with the chemical formula, means same connectivity. They are connected in the same order, right? And if you remember from the definition of the stereoisomer on the previous slide, same formula, same connectivity, but the different orientation in the three-dimensional space. So let's try to make an analogy with that. So if you try to make an overlap of your left hand on the top of your right hand, can you overlap? No, because if you try to overlap, you will always end up with one thumb of the one hand on one side, and the other thumb from the other hand is going to be on the opposite, like 180. So you will not get a perfect overlap of your left and right hand if you try to. So why you cannot do an overlap? Because they are not same. Your left and right hand are different. And why they are different? Because they are mirror images of each other. And what are mirror images? So mirror images means if you take a mirror and if you put your one of the left hand in front of the mirror, you will see an image over here, right? And whatever the image you are seeing of your left hand, that image is exactly like your right hand, right? So that mirror images is normally is same as a right hand image, right? And then if you try to overlap your right hand with the left hand, again the mirror images is not going to overlap with each other perfectly. So that means that object is a chiral object. So let's go to the definition of the chiral object. Chiral object or molecule have non-superimposable mirror images. Means you cannot superimpose the actual molecule or the hand in this case with their mirror images molecule. You cannot overlap, right? So make sure you know this definition, which is non-superimposable mirror images, means they don't have any symmetry, right? And we call those kind of molecule and object are chiral objects. So that means our hands are chiral, they are not symmetrical. We can say chiral, right? And can you give me one more example of chiral object? Our left hand, right, like shoes, they are also chiral, they are not symmetrical. We cannot put our left foot into our right hand or the right shoes, or and vice versa, right? So that's why the shoes are also a chiral. Now let's talk about the achiral one. What is achiral molecule? The term A means non, right? Not a chiral molecule, are superimposable to their mirror image. It means those molecular objects that has a superimposable mirror images are called achiral, means they have a symmetry. For example, if I put a glass in front of the mirror, you will see an image of the glass over here, right? This is an image, and if I try to overlap this image on the top of the original glass, you will always perfectly overlap, right? Because there is no problem with it, right? So that means the mirror images are superimposable. So that means glass is achiral, right? So just remember from this topic that achiral object or molecule are those objects that have non-superimposable mirror image, means no symmetry, and then achiral molecules are objects that have superimposable mirror images, right? That means they have symmetry. So this is the concept that you're gonna learn from this slide. Now let's talk about one more term which is important in the carbohydrate, right? In stereoisomers called enantiomer. So enantiomer is a pair of non-superimposable mirror images, right? If you remember on the previous slide, our hands are non-superimposable, left hand, right hand. So if I put our left hand in front of the mirror, I'll get a mirror image. I'll get the image of the right hand, right? So the left hand and its mirror image are enantiomer because they are a pair, right? Similarly, if we take an example that is given over here, one of the molecule, if it has a mirror image, and if I take this molecule and try to overlap on our original molecule, they are not going to overlap, means they are non-superimposable. That means they are chiral molecule, right? And then if they are chiral molecule, they cannot overlap with each other, they are mirror images of each other. So I can say this thing is a mirror image of this, or I can do say a vice versa, this structure is a mirror image of this structure, right? So we can say both of them are mirror images of each other. So we can also say these are isomers, enantiomers. So enantiomers, right? So to get an enantiomer, you need at least two molecules, right? You cannot say this molecule is an enantiomer without including this molecule, right? The mirror image. So just know this concept, and then let's go to the another slide, and then we're gonna talk about the molecule that show this kind of enantiomer or chirality, right? Now we're gonna talk about the chirality center, right? So what a chirality center? Chirality center is a tetrahedral carbon bonded to 4 different atoms of group of atoms. This is an important definition, make sure. So chirality center is a carbon that is bonded to 4 different atoms or molecule. And if the carbon is bonded to 4 different atoms, that means this carbon is a tetrahedral. Geometry. And this carbon we call as chiral center, chiral carbon or chiral center, right? So let's take an example over here, right? So we have 2 examples. This is 1, this is 2, and if you see this carbon, right, this is a line angle formula. So this is a carbon, this carbon is attached to one of the group CS3, another one is hydrogen, third one is ammonium NH3+, then the fourth group is this. So I can see this carbon attached to 4 different groups, CS3H, NH3+, and C double bond O negative, means this carbon is a chiral carbon. Right? If it's a chiral carbon, that means this molecule where you have this chiral carbon present, that molecule is a chiral molecule. right? Because you have a chiral carbon, that makes the molecule as a chiral, and if the molecule is chiral, that means you can have a non-superimposable mirror images of this molecule. So if I take a mirror and if I put in here, so these two are mirror images of each other, right? And they are non-superimposable, so that means they are enantiomer of each other. So this is how you can just follow the concept, and then you can easily say whether the molecule are chiral. If yes, whether they are both the molecule that is shown over there are enantiomers or not. So the requirement is you need to have at least one carbon in that molecule as a chiral carbons before you can say that molecule is a chiral, or they can have a enantiomeric mirror images, right? So let's try to do one problem based on the chirality, and let's try to apply the concept that we just learned. So what is chirality center? Chirality center are that carbon that is bonded to 4 different groups or 4 different atoms. Circle any molecules that are chiral and identify the chirality center with an star mark, right? So let's start with the A. So let's see how many carbon atom you have in here. You have 1 carbon, 2 carbon, 3, 4. Out of the 4 carbon, which carbon is a chiral carbon? So what's the definition of the chiral carbon? That carbon has to attach with 4 different groups. If you talk about the first carbon, it is attached what? 3 hydrogen. The time I see 2 of them are same, this carbon cannot be a chiral carbon. What about this one? Again 2 hydrogen. Same atom. Out of the 4, 2 are same, not a chiral. Again 3 hydrogen. Out of the 4, 3 are same, not a chiral carbon. But if you look around this carbon has this one of the group, second is hydrogen, third group is this CH3, and fourth group is this 2 carbon. So this carbon has 4 different group attached to 4 different bonds. So that means this carbon is your chiral carbon or chiral center, right? Let's do the second one. This carbon is not a chiral carbon, this carbon is not a chiral, this carbon is not a chiral. We already explained why. But this carbon is a chiral because you have hydrogen, you have CH3, second different, then you have now. So if you see, this carbon also is not a chiral carbon. Why? Because you have CH3, right? What's this thing? This group is also a CH3. You have 1 carbon and 3 hydrogen. So this one and this one are same group. So out of the 4, 2 are same. B is not a chiral molecule because it doesn't have any chiral carbon in there, right? Let's talk about the other one. What about this carbon? No, because it has 2 hydrogen, same, not a chiral carbon. What about this one? You have oxygen, one of the group is this CH2OH, second group is CH2OH, same group, not a chiral. It doesn't have any chiral carbon in there, right? What about the D? Let's talk about the D. This carbon is a chiral? No, because it has 2 hydrogen, same out of the 4. What about this? You have 1, 2, 3, you have hydrogen, and then this double bond means you have same bond attached to oxygen, same atoms, not a chiral carbon. But what about this one? You have one of the group OH, second group hydrogen, the third group is CH2OH that is connected with this carbon, and the fourth one is C double bond O. So that means this carbon is attached with 4 different groups or atoms. So that means this carbon is a chiral carbon or center. So this is how you normally differentiate between a chiral center or a achiral center, right? You have to just look for how many different atoms or groups are attached for that particular carbon. If you have 4 different groups, that means you can say easily that that carbon atom is a chiral center. right? And that molecule become a chiral molecule later on. So that means this molecule, entire molecule, is also a chiral molecule, right, because of the presence of the chiral carbon. But it's not always the case, we'll talk about later on. But if you have any chiral carbon, you can assume that molecule is a chiral molecule, right? But this is not always the case, right? But mostly this is the case. So now we're going to learn about the Fisher projection. What are Fisher projection and why we use them? So Fisher projections are the models that are used to draw a molecule with chirality center. So any molecule with a chiral center, we can draw that molecule with the help of a Fisher projection, right? In Fisher projection, the intersection of the horizontal and vertical lines normally show a chiral center. If you have a vertical line and horizontal line where they intersect, this represents a carbon, and this is most probably is a chiral carbon, right? So if you see any this kind of structure or of a molecule in the form of horizontal and vertical line crossing intersect with each other, that means we are drawing a Fisher projection for that molecule, right? And if we talk about the enantiomer, how you can draw an enantiomer using a Fisher projection, that means opposite horizontal atom at all chirality center. Let's take an example. What does it mean? Let's take this example over here. You have a horizontal and vertical lines, then you have 4 different group attached to it. One different group, second, CS2OH, H, all 4 are different. So that means this carbon is a chiral, right? Because the intersection one is a chiral. And then if I put a mirror over here, then you will get a mirror image. In mirror image, you will get the OH on the other side and H on the other side, right? And then this is a chiral center. So these are enantiomer of each other. And what you can see in the enantiomer, so all the atoms or groups that are present in the enantiomer, they are on opposite side if you compare the mirror images of each other. So if this is your actual molecule and this is your mirror image, you will see the OH is placed now on the opposite side, right? So it doesn't matter how many chiral center you have in that molecule, if all the chiral center has the atom or group on the opposite side compared to the original molecule in the mirror image, that means those two molecules are enantiomer of each other, right? We'll take some example later on, but this is a concept that you need to understand about this right now. Now let's try to make a correlation between the skeletal line that we just learned on the organic chemistry part and a Fisher projection, how they are different. So Fisher projection shows carbon chain vertically, something like this, right? So if we already learned from the previous slide, the intersection is a chiral carbon, right? Whereas the skeletal line structure show the carbon chain horizontally, so something like this, that we learn in the organic chemistry part. So over here you have the carbon vertically in the Fisher projection, in the skeletal line you have horizontally. These are your carbons, right? So this is how you can differentiate whether we are talking about the Fisher projection or a skeletal line structure in organic chemistry, right? So there are a couple of things that you need to know in the Fisher projection. First, rotate skeleton structure to make the vertical chain vertical and place most oxidized carbon near the top. So whenever you're making a Fisher projection, then whatever the carbon is more oxidized, you have to put on the top. So this carbon has to be more oxidized carbon, right? And if you remember, what are the 2 different groups that we normally use in a carbohydrate? One is ketone, second one is aldehyde, right? So if you have, so which one is the more oxidized one? The carbon that has a carbonyl group, and this carbonyl group is present in ketone as well as in aldehyde. So that means this carbon has to given the preference to be on the top if there is a possibility, right, in the feature projection. Then carbon-carbon bond project away from the viewer, then vertical dashed bonds. Horizontal bonds project towards the viewer, wedge. So we have two kind of bond, dash and wedge. Dash means that bonds are going away from the side, right? Wedge means they are going towards you. We're gonna see it in the next slide. What does it mean, dash and wedge? And only chirality center are shown as an intersection, right? So these are the intersection, and only the intersection carbons are chiral carbon in feature projection, right? And all the groups are shown in condensed forward. So whatever the groups is attached on this, you always show in the condensed form, right? So we're gonna learn about all these four rules on the next slide, right? You can see. So let's take an example over here that we just learned on the previous slide, the four concept. Then the first one is wedge and dash notation. So if you can see, this is the molecule, right? This is a line structure formula, and you have a wedge and dash, right? So that dash means is going away from the viewer, and then wedge means is going towards the viewer, right? And this is the carbon chain. But if you rotate the carbon chain and try to put it in a straight line, then the most oxidized carbon has to be on the top. So out of all of them, which one is the most oxidized carbon? This carbon, because it has a carbonyl group, C double bond O. So this carbon is gonna be on the top, right? Then this is second, this is third, and then on the second carbon you have OH and H, OH and H. On the third one you have OH, right? Third one you have OH and two hydrogen. So if you remember, in feature projection, that the most oxidized carbon should be placed on the top, and then you will have the vertical cross section line. So this carbon is a chiral carbon, not the first one, not the third one. Carbon 1 and 3 are not chiral, right? And then if you try to put this three-dimensional way of drawing the structure, because we have wedge and dash, because we normally show wedge and dash in a three-dimensional form, and if you project in the 2D, you just put a horizontal and vertical lines. So this cross section is your chiral carbon. So let's see how many carbon you have in there. You have 1, you have 2, you have 3. Out of the 3 carbon, which one are chiral? So the first is not a chiral carbon, right? Because you have C double bond O, you have 2. So if you have anything like this, C double bond O, and then you have H and R. So in feature projection, if you have a double bond, we assume you have a single bond and both of them attached with the oxygen, right? Double bond is, you can treat that each bond is attached with the separate oxygen. So that means 2 same atom. This carbon is not a chiral carbon. Second one is a chiral because this carbon is attached to 4 different groups, right? But the third one is again is not a chiral. Why? Because this carbon is attached to 2 hydrogen atom, 2 same kind of atoms. So that means this is not a chiral, because by definition carbon has to attach with the 4 different groups. So this is a feature projection, and only the intersection carbon is a chiral carbon, right? Now let's try to learn one of the concepts that we haven't done on the previous slide about the L form and D form for any carbon that is chiral, right, in a molecule. So let's take an example over here. So let's say out of the 3 carbon, 1, 2, 3, because we know that the intersection 1 is a carbon, right? The first carbon is not a chiral carbon because you have 2 oxygen, same type of atom or group attached out of the 4. The third one is also not a chiral carbon because out of the 4, 3 are same, right, CS3. But the second carbon is your chiral carbon because on this carbon you have 4 different group attached, right? Now if you talk about the horizontal line, you have an OH and hydrogen, right? But can you draw the same structure, something like this, where you have the hydrogen on the left side and OH on the right hand side? Yes. So how would you differentiate between the 2 types of compound? In this case both of them are lactic acid, but how would you differentiate? Because they are stereoisomers, right? Because you have a chiral center, this is a chiral molecule. So you have to differentiate between the two by a form called L form or D form. So you have to look for the hydroxyl group, OH group, that is present on the chiral center. If that OH group present on the left hand side, then the form is L form. If the OH on the chiral center present on the right hand side, then this is a D form. So you can easily figure it out or easily name the molecule either as D and L. So if let's take an example. If I take a mirror, put a mirror in between the two molecules, so that I can say they have the chiral carbon atom. So this entire molecule is a chiral molecule. If the molecule is chiral, they can have a mirror image which are non-superimposable. If you take this entire mirror image and if I try to put it or superimpose on the top of it, they cannot. So they are non-superimposable mirrors. So that means, and the chiral atom, carbon atom, whatever the is attached on the chiral atom, we have H and OH, they are on the opposite side in the mirror images, right? So that means they are enantiomer of each other, right? So in this slide we learn about what is D and what are L, and then what are enantiomer one more time, and what are chiral molecule, right? So if we already given with the L lactic acid, you can form a D lactic acid, right, by just moving the OH and H on the opposite side, because this can be your mirror image, right? So in the previous slide we just learned about what are L isomer or D isomer for any feature projection. So let's take an example one more time over here. If I have CS2OH and then you have H and OH and then you have CS3. So which kind of isomer is this one, this feature projection? So in this feature projection, this is your chiral center, and on chiral center, if the OH is present on the left hand side, that means this is a L isomer, right? And if I put a mirror in front of it, then you're gonna get this kind of structure as a mirror image. You get H on this side, OH on the other side, and then CS3 on this side. So which kind of isomer is now this one? So this is a chiral center one more time, because why this is a chiral? Because you have 4 different group attached to this carbon, and in this chiral center OH is present on the right hand side. So this is a D isomer of the molecule, and they are non-superimposable mirror images of each other. That means they are enantiomer too, right? So this is how you can determine. But if I give an example where you have multiple chiral carbon, in this case we had just one, but if I give you this example over here where you have C double bond O H, then you have 2 chiral center, and at the end you have CS2 OH, you have H OH and H and OH. So which kind of isomer is this one, L or D? Because we just learned about, you have to see the chiral center. So this is your one of the chiral center, this is your second chiral center, this is not a chiral center, this is not a chiral center, right? Because out of the 4 carbon bond, 2 of them are same. So these 2 carbon are not chiral except the one that we have on the intersection, right? So now how are we going to figure it out whether this is the L form or D form? Because you have 2 OH group on 2 different chiral carbon, one is on the left, one is on the right. So what should be the name, L form or D form? So you always have to look for the chiral center which is furthest away from this oxidized carbon, right? So whatever the oxidized carbon that you have in that feature projection, you have to look for the furthest chiral center. So this is the furthest from this, so you have to look the OH group location on the furthest chiral center. In this case this is on the right hand side, so this is your D form, right? But if I make a mirror in front of it, this is your mirror, and if I need to make a mirror image, it's gonna be the same thing, right? Then H is gonna be on this side, OH on this side. Then you have OH on this side, H on this side, then CH2OH. So now this is again the 2 chiral center, and this chiral center is again furthest from this carbonyl group or the oxidized carbon, but in this case the OH is placed on the left hand side, so this is your L form, right? And then are they mirror images of each other? Yes. Are they non-superimposable? Yes. They are not superimposable, so they are enantiomer of each other. So just remember again the definition of enantiomer. Enantiomer are the mirror images of each other where the group attached to the chiral center are both on the opposite side. They just switch their position. So you have 2 chiral center, and in both the chiral center now the atoms are arranged oppositely. Over here in the first, the OH is on the left hand side. Over here now it goes to the right hand, and the second chiral center, OH was on the right hand side, now over on the left hand side. So that's why they are enantiomer of each other, because each chiral center has now atoms or group attached in a different location, right, left or right. So this is how you can determine also D and L form if you have multiple chiral center, right? So we already covered all the concept, enantiomer, D or L form, how you're going to make a mirror image, right? So make sure you remember this kind of concept. Now let's talk about the physical and pharmacological properties of the enantiomer. So enantiomer have identical physical and chemical property, means because they comes from the same molecule. That's why they have the identical physical and chemical properties, and that's why you cannot differentiate or you cannot separate these enantiomer, right, either physically or chemically, but you can still make a difference between the two on the basis of the chiral environment, right? So let's take an example over here. If we take a chiral drug, what is chiral drug? Means you have 2 different forms, right? One form and second form, D and L form, that we already discussed on the previous slide. Let's take, and then you have a receptor on the cell membrane, right? If you take that drug, and then if you have L isomer, then the receptor and the drug make a perfect contact, and then you will get the desired result that you wanted, right, by taking that drug. But if you take a D enantiomer of that drug, and if you see the receptor won't change its surface. If you have a different drug with a different structure, or you can say a mirror image of that drug, then it won't be perfectly matched with the receptor on the surface of the cell, and then the cell will reject this drug, right? And then the receptor will recognize this drug as a different compound, right? And then that's why the chiral compound has different pharmacological effect, because different receptor attached with different kind of enantiomer, right? So this is why you have to have the right kind of drug with the right enantiomer, right, if you have to have any kind of effect by taking that drug. Now let's talk about one of the topic called racemic mixture. Racemic mixture are the mixture where you have the equal proportion of both the enantiomer of the drug. Means you have 50% of L form, 50% of the D form, right, when you mix both of them in equal amount. And we use this drug when both the enantiomer of our drugs are safe, right? Whether the L form or D form, both of them are safe, we can make a mixture. Or if one of them is inactive, so it wouldn't do any harmful effect, right? It wouldn't do anything. For example, ibuprofen. You have S-ibuprofen is an analgesic where the R-ibuprofen is inactive. Means what is this S and R? This is another way of writing the iopic name for the enantiomer. You can have S and R. We are not going to go in detail what is S and R, but this is like L and D, right? So if you have ibuprofen is a racemic mixture where you have S-ibuprofen, which is the main component of the drug, and R ibuprofen is inactive, so we are using as a racemic mixture. Now let's talk about how you going to differentiate the enantiomer that we have in a solution, right? Whether it's L form or D form, and if both the form are available, then which form is present in high amount, right, in more ratio. Because enantiomer have same chemical and physical property, we cannot differentiate on the basis of chemical and physical separation, but we can use a special concept. You can say specific rotation to differentiate how much ratio of each of the enantiomer is present in that mixture, right? And what is a specific rotation? A specific rotation normally tells you how much rotation you can get for the plane polarized light. What is rotation? Let's take an example. If you have a light source, right, and if the light source is going in every direction, right, and if we put an object with a slit in here, and if you shine that light on that slit, then which kind direction of the light is going to pass through the slit? So the light that is going parallel to the slit is going to pass through the slit, and all other light source or the direction of the light is going to block by that slit, right? So only one kind of light is going to pass through the slit, which is parallel to the slit, right? And if this is called as plane polarized light, and if I pass this plane polarized light through a solution, that is a racemic mixture. That is a mixture of both enantiomer, right? Both the enantiomer, and we don't know how much amount of each of the form, L form or D form, right? The enantiomer. Then if the plane polarized light pass through this solution, and if it tilt clockwise, something like this, then we can say that enantiomer is a dextrorotatory enantiomer, or in the form of plus or small d. If the plane polarized light come out from the solution as anti-clockwise, something like this, then we can say that one is a levorotatory enantiomer, or we can represent in the form of a negative or small l, right? So we only talk about this thing, how we differentiate between dextrorotatory and levorotatory. So the enantiomer that rotate the plane polarized light clockwise, that is dextrorotatory. If that enantiomer rotate the light on counterclockwise, that enantiomer may be called as levorotatory, negative and L. And if that solution has a racemic mixture, means equal amount of both the enantiomer, what do you think? The plane polarized light is gonna go clockwise or counterclockwise? It's not gonna go anywhere, because one of the isomer is trying to move the plane polarized light on anti-clockwise, but the other wants to go to the clockwise. So there is a equal force on both direction, then there won't be any net effect. So the light will come out to be exactly as straight, whatever way it entered, right? So that means that optical mixture is optically inactive. Why optically inactive? Because it didn't rotate the plane polarized light in either direction, right? So just remember from this topic is, if the plane polarized light is passing through an enantiomer mixture, and if it turns clockwise or anti-clockwise, you can have dextro levo, or we can say that solution is optically active. Right? How would I know this is optically active? Because something happened to the plane polarized light, right? It changed its direction. That's why it's active. That solution is working. But if it's a racemic mixture, the solution, the enantiomer is still working, but the net effect is 0. That means the solution is optically inactive. And our achiral compound are optically inactive because achiral compound doesn't have any enantiomer, right? They don't have non-superimposable image. That's why they are optically inactive too. Yeah. Now let's talk about one of the term called diastereomers, right? So what are diastereomers? Diastereomers are the stereoisomers that are same at one or more chiral center, but not all chiral center. So this is the important term, not all chiral center, right? So let's take an example over here and then try to learn what are diastereomers, right? So let's take, this is a feature projection of one of the carbohydrate. How many chiral center you have in here? 1, 2, and the name is erythrose. And then if I put a mirror in front of it, then you get a mirror image over here. Then what's you have in the mirror image? Then in all the chiral center, the groups of atoms are arranged are already flipped. They're already on the other side, right? In both of them you have 2, and both of them you have OH on the other side and hydrogen on the other side, right? So that's why they are enantiomer. But if I take this molecule over here, what's the difference between this and this molecule? They have the same molecular formula, but on out of the 2 chiral center, only one of the chiral center has the atoms where it get changed, the groups, right? Now the OH is on the left hand side, but over here is the right hand side, but the other chiral center atom doesn't flip, is on the same side, right? So they are diastereomers, right? Because it wouldn't change both the chiral center, right, atoms. If we take about now this molecule and this molecule, what's happening over here? Again, on the first chiral center, the atom or group is flipped. Now they're on the other side, OH. But if you can see the second chiral center, the molecule or the groups are same. So only out of the 2, only one, not all of them, only one is changed or flipped. That means they are diastereomers, right? But if you say this example over here, now in this case both the chiral center flip their atom or groups, right? Now both of them in opposite direction, OH on the other side, OH on the other side, right? So they are enantiomer. If we compare the opposite, they are diastereomer, they are diastereomer. So just remember the concept. If you have any number of chiral center, and if you have another molecule with the same molecular formula but on the chiral center, if all the chiral center doesn't flip their atom or group at the same time, they are diastereomers. But if all the chiral center flip their atom on the other side that we have on the original molecule, then they are enantiomer. So just remember this concept. Let's say how many maximum number of isomers you can make for a molecule with n chiral center, right? So let's say if you have just one chiral center, how many stereoisomer is possible? 2 power 1, 2. If you have 3 chiral center, then 8 stereoisomers are possible, whether it's an enantiomer or diastereomer, right? By combining all of them possible isomer, we can have 8, right? Glucose has 4 chiral center, and therefore there are 16 possible stereoisomers of glucose. 2, 4 is 2 times 2 times 2 times 2 is 16. So 16 stereoisomers are possible. So make sure you remember the formula 2 raised power n. Now let's try to do one problem based on the concept of enantiomer and diastereomers. The problem says indicate whether each of the following paired stereoisomers are enantiomer or diastereomer. Let's take this first one. So you have how many chiral center? 1, 2, 3, same. 1, 2, 3, same molecule. But if you can see on the first chiral center, you have a group in opposite direction, right? You flip. On the second chiral center, you again flip, right? On the third chiral center, you again flip. OH goes that side, H goes on this side. So they are enantiomer. So the definition of enantiomer is if all the groups on every chiral center flip at the same time, we call as enantiomer. Let's talk about other two, the B part. The first chiral center, second chiral center, third chiral center. The first one hasn't flipped. Second one, it flipped in this case. Third one hasn't flipped. So out of the three chiral center, only one chiral center flip its atom or group, right? So that means they are diastereomers because all the chiral center didn't flip at the same time their atom or groups. Let's talk about the C. You have three chiral center. One more time, the first chiral center flip its group or atom. Second chiral center also flip. You have OH on this side, OH on this side. The third chiral center didn't flip. They're on the same side, OH, OH, right? So out of the three, only two changed, but the third didn't flip. They are diastereomers. But because of for enantiomer, all of them has to flip at the same time. So this is how you can differentiate between enantiomer and diastereomers. Now let's go to this other topic, which says the structure of the common monosaccharide. So in this topic, we're going to talk about different structure of the monosaccharide and how you're going to name it, right? So there are three structural characteristics that you must include, you must know. First, the number of carbon atom in that particular monosaccharide, right? If you have a 3 carbon, then the name is triose. If you remember, the ending word of the carbohydrate end with OSE, O's. If you have a 4 carbon monosaccharide, tetrose, 5 carbon, pentose, 6 is hexose. So depending on how many carbon atom you have in that particular monosaccharide, you can say whether it's a triose, tetrose, pentose and hexose. Second, if you remember, every carbohydrate has either aldehyde or a ketone in that structure. So if an aldehyde is present, then we call as aldose, and if a ketone is present, then we call as ketose, right? Then the last one, the configuration of each chirality center, whether the OH is present on the left or right, depending, we can have D or L isomer, if you remember, right? So we're gonna use all these three information and try to name the molecule of the monosaccharide, right? Let's go to the next slide and then we'll try to do some example. So let's take an example over here. This is a Fischer projection of one of the molecule called L-glyceraldehyde. How many carbon you have in there? 1, 2, 3. 3 carbon is triose. And then what kind of group you have in there? Aldehyde. So aldotriose. So this L-glyceraldehyde is your aldotriose. And why this is L? Because the chiral center configuration, the OH is on the left hand side, so this is L. Why this is D? Because this OH is on the right hand side on the chiral center, so this is L or D, aldotriose. Let's take an example, one more. One of them is fructose. This is one of the monosaccharide. Right, we're going to talk about the other two monosaccharide later on, but just remember fructose is one of the monosaccharide, and fructose is a ketohexose. Keto means it should have a ketone group, and hexose means 6 carbon. So let's say this is a structure, Fischer projection for fructose. So you have 1, 2, 3, 4, 5, 6. Out of the 6, how many are chiral center? This carbon is not a chiral center because attached to 2 hydrogen, same thing. This carbon is not a chiral center. Then this carbon is a chiral, this carbon is a chiral, this carbon is a chiral, this carbon is not a chiral. One more time. Why? Because you have carbon and 2 hydrogen atoms, same kind of group. But if you can see, the third one is a chiral because you have OH, you have H on the top, you have a different group, on the bottom you have different group, so 4 different groups. Same thing apply for 4 and 5. All 3 are chiral center. But how would I know whether, so which kind of this is keto hexose. We already talked 6 carbon ketone group, keto hexose. Then how would I know this is D? So which one is the most oxidized carbon? This one is the most oxidized carbon in this molecule, and you have to look for the furthest away chiral center. This is the furthest away from this oxidized group. Then you have to look the OH configuration, where you have on the left or right. In this case is on the right, so this is why this is D. So D fructose. Right. Let's talk about the other 2 monosaccharide. One of them is a glucose, second one is your galactose, right? So these 2 are also monosaccharide. So basically you have 3 different types of monosaccharide: glucose, galactose, and fructose, right? So let's see what you have. So glucose has how many carbon? 1, 2, 3, 4, 5, 6. Galactose again 1, 2, 3, 4. No, 1, 2, 3, 4, 5, and 6. Both of them have 6 carbon. Which kind of group you have? Functional group, aldehyde. So I can say this is aldohexose. Both of them, right? 6 carbon, aldehyde, both of them. And then why this is D? Why this is D? Because the last chiral center has OH group on the right hand side, last chiral center OH on the right hand side, D. But if you see both the structure, they are almost similar, but the only difference is on the carbon number 4. What's the difference? In carbon number 4, the OH is placed on the right hand side in glucose, but in galactose that OH is placed on the left hand side. That's the only difference in the structure between glucose and galactose, right? So if you remember the structure of glucose, that means you already remember the structure of the galactose. You have to just flip the OH on the 4th carbon, right? And we can say that glucose and galactose are diastereomers. What are diastereomers? If you have at least one of the chiral center flip their atom or group, right? So except the 4th, everything is same, only the 4th is flipped, so they are diastereomers, right? So yeah, so just remember the concept, and then what are the different kinds of monosaccharide we have, right? And then all this information, diastereomers, right? What are aldohexose, aldoketose, right? So let's talk about D sugar. We already talk about what are the different types of sugar we have. We can have D or L depending on the OH group on the last chiral center furthest away from the most oxidized group or the carbonyl group, right? We already talk about this thing. So I hope everyone already know till now that how you differentiate L and D carbohydrate from the structure, Fischer projection structure. Let's take a couple of examples. Why every carbohydrate molecule in the Fischer projection are D? Because the last chiral center further away from the oxidized carbonyl group, the OH is on the right hand side. That's why all of them are D, right? Now let's try to do this problem based on the concept that we just learned till now. So the problem is saying answer the questions below about the following aldopentose. Aldo means aldehyde, pentose means 5 carbon produced in nature. So how is D-arabinose different from D-lexose, right? All of them are aldopentose, right? And then it says how D-arabinose and D-lexose or lexose are different. So arabinose and this one, right? So let's say how many chiral center you have in there. You have 1, 2, 3, same thing, 1, 2, 3. On the first chiral center, the atom or groups are flipped, the configuration are different, okay? The second one, again the configuration are different, but on the third one the configuration are same. That means out of the 3, only 2 have changed their configuration around the chiral center, that means they are diastereomers. You can say diastereomers, they both of them. Let's do the second one. Are these monosaccharides D sugar or L sugar? So let's talk. The last one has an OH on the right hand side, so this is a D, and this one on the right hand side D, so both of them are D sugars, right? How many chiral center do these sugar have? So how many chiral center? 1, 2, 3. So they have 3 chiral center. What is the maximum number of stereoisomers? If you can remember, it's 2 raised power n, that is 2 raised power 3, so 2 times 2 times 2, that is 8. So you can have 8 stereoisomers, right? So you already have 4, you are just need to make 4 more in the form of L isomer, right? So once you made that 4, then total is 8. So this is how you normally solve the problem, based on the L form, right? How many isomers, how many chiral center you have it. So now we're going to talk about the ring form of the monosaccharide. So till now we just learned about the Fischer projection, which is an open structure for the monosaccharide, right? And so let's talk about the ring form. Why the monosaccharide mainly stable in the ring form compared to the open form, which is a Fischer projection, and why is the case? So whenever you dissolve any monosaccharide in aqueous solution, the hydroxyl group near the end of the chain of pentose and hexose react with the carbonyl carbon to form a stable 5 or 6 member ring, right? So we'll see on the next slide what does it mean. We'll do some example how to draw it, but right now just focus on this thing. Those monosaccharide that has 5 or 6 carbon can form a ring form, because smaller rings are less stable and they do not form. So you need to have at least 5 or 6 carbon before the open form convert into a ring form. And ring form is a stable form if it's made up of 5 or 6 carbon, right? So let's say if they make a 6 member ring with an oxygen at one of the atom and 5 are carbon atom, we call those kind of structure as pyranose. If they make a closed cyclic ring like structure with 5 member ring in which one of them is oxygen and another 4 are carbons, we call as furanose. So we can make something like this. Pyranose is a 6 member ring, so 1, 2, 3, 4, 5, 6th one is oxygen. Then furanose is a 5 member ring, is something like this. 1, 2, 3, 4, and 5th is your atom, oxygen atom, right? So this is your furanose, this is your pyranose. So let's go to the next slide, and then we'll try to learn how to convert a Fischer projection, the open form, into a ring form. And before we go into the next slide, we're gonna tell you what's the name of that ring form of monosaccharide. We have a special name we call as Haworth projection, right? Like we have a Fischer projection where you have the vertical and horizontal line. Similarly, the ring form or the cyclic structure form we call as Haworth projection. So make sure you know the difference. Haworth is a closed ring structure, Fischer is an open-ended structure. So let's go to the next slide now, and then let's try to learn how to make an Haworth projection from a Fischer projection. So now we're gonna learn about how this ring form normally form. We take an example of D-glucose, right? So in D-glucose, according to the rule that we learned on the previous slide, it says the last chiral carbon atom that has an OH group, the hydrogen from this OH is going to get attached with this oxygen, right? And then because attachment of the hydrogen on this oxygen, then this double bond is gonna remove, and now this carbon has only 3 bonds, right? One with oxygen, one with hydrogen, one with this carbon on the bottom. Then this oxygen which is left after the hydrogen is removed is going to combine with this carbon over here, right? And this already become OH, right? Because this hydrogen is attached with this oxygen. So if you see carefully over here, you're gonna get a cyclic structure. Start from 1, 2, 3, 4, 5, and the sixth one is oxygen. So you're gonna make this cyclic form. And if you have a 6 atom, the Beck's form is a hexagonal. So this is your hexane. You have this oxygen, which is this, right, attached with this carbon number 1. This is your carbon number 1, right? And then the OH group on that carbon is this, and the hydrogen is this. And for each carbon, for the remaining thing is the same thing, OHH, OHH. We're gonna talk about this later, why the OH is on the bottom, this OH is on the top, somewhere like this. But this is a basic concept, how we're gonna make the 6-member ring with an oxygen as one of the atom, right? And then we already talk about this is the bond that is formed new between oxygen and carbon on the first one, right? And where does this 6th carbon goes? 6th carbon attached to the 5th carbon, which is a part of the ring. This is your 5th carbon, and then it goes CS2OH on the top, right? So in the last slide we just learned about how the Fischer projection you're going to convert into a cyclic ring structure. But the important thing in the cyclic ring structure is you're creating a new chirality center when you make a ring form, right? So if you remember from the D-glucose, out of the 6 carbon, how many chiral center we had? We had only 4 chiral center. The first one is not a chiral, the last one is not a chiral, so you have just 4 in intersection in there, right? So glucose had 4 in the Fischer. But when you're converting into a cyclic form, now how many chiral center you have in there? Let's see. This is your chiral center, this is your chiral center, this is chiral, right? But now you have one more chiral, which is this carbon, that is formed by a formation of a new bond between oxygen and carbon number 1. So why this is a chiral center? Because on this carbon you have one hydrogen, one OH, one of this group, another is this group. So you have 4 different group attached on this carbon. So this is a chiral carbon, right? And we call this a newly formed chiral carbon as a anomeric carbon or anomeric center, right? And then what's special about this anomeric center? The OH group and hydrogen on this anomeric carbon can be flipped. It can be present on the top or on the bottom, or vice versa, like in this case, right? So it can, if the OH is present on the bottom on this anomeric carbon, we call this as alpha isomer, and if the OH is present on the top, we call as a beta in the cyclic form. And then this D comes from the Fischer projection. From which Fischer projection we make this cyclic structure? From the D-glucose Fischer projection. So make sure if you have beta D or alpha D, alpha beta comes from the cyclic form and D comes from the Fischer projection, from where we got this cyclic form, right? So on this slide we're going to talk about the same alpha ionomer and beta ionomer, because present the anomeric carbon. If OH is present below, then is alpha. If OH is present above, then cyclic structure, then beta. And the important thing over here is both alpha and beta isomer are in equilibrium with an open chain in aqueous solution. So if you put this cyclic ring structure in water, they are flipping between Fischer projection and cyclic ring structure. So they are flipping between each other, right? But most of the time they may be present in the cyclic form because they are the most stable form for any 6 carbon ring, right? but they are in equilibrium. Sometimes they present in the Fischer projection, another time it convert into a cyclic form, right? So the reaction keep on going. So if you dissolve a glucose in a solution, you will see 90% of the time they are present in a cyclic form, and another time it present in the Fischer projection, but mostly glucose would prefer to be in a cyclic form. Now let's take an example of fructose, another kind of monosaccharide, and then try to learn how it makes a 5-member ring, which is a furanose ring. So if you see this structure over here, you have 6 carbon. Out of the 6 carbon, only 3 are chiral center, right? And then according to the rules, the hydrogen on the last chiral center over here is going to attach with the most oxidized carbon, oxygen. So this hydrogen is going to remove from here and is going to attach on this carbon, oxygen. The time it attach, this bond is not required because oxygen already got the bond that it needed. Now carbon has only one bond with oxygen, one bond with the carbon on the top, one bond with the carbon on the bottom, right? So how many bond carbon has? 3. It need 4. Then the oxygen says, I can attach with you because I already lost one of the bond in the form of hydrogen, so it just bind with this carbon, right? And the time you see this thing, you will see now you have the closed ring, this. And how many atoms you have in this closed ring? 1 2 3 4 5. Out of the 5, 4 are carbon, 5th one is hydrogen, 5th one is oxygen. So the best possible way is oxygen, and then you have 5, right? 5-member ring. And then on the second carbon you have what? An OH. This is your second carbon where the oxygen is attached, and on the second carbon you have the first carbon attached, which is this one, right? So this is how you have the first carbon is not the part of the ring because the oxygen didn't attach on the first carbon, it attached on the second carbon, so that's why. So again the same thing, if the OH is present on the bottom of the ring, then is alpha. If it was on the top, then is beta. And why this is D-glucose fructose? Because it comes from the original D-fructose Fischer projection, right? So it can make both of these cyclic structure, right? And they are in equilibrium. They can keep on flipping between each other, but most of the time they want to be remain in the cyclic form. So now let's take an example how to draw a Fischer projection into a Haworth projection, which is a ring-like structure projection. So let's take an example of glucose. Okay, so this is your D glucose. How would I know this is a D? Because this is the last chiral carbon out of those 4 that we have, and the OH is present on the right hand side, so this is D glucose. How are you going to convert this Fischer projection into a cyclic structure? So first, the firstmost thing is just remember all the atoms or groups that is present on the right hand side that goes below the ring in the cyclic structure. Whatever the group is present on the left hand side, it will go up on the ring structure, right, on the top. So let's now try to learn how to convert a Fischer to a Haworth. So the first step is take the hydrogen on the hydroxyl group from the last chiral center. So take it out this hydrogen and put it over here. So I am removing this hydrogen from here. The time the hydrogen goes onto the top oxygen, then this one of the bond oxygen needed is not required anymore, right? So it's done. And the time You remove one of the bond. This carbon has 3 bond now. It need 4, and this oxygen has just 1. It needed 2, so they can come together and make a bond with each other, right? And the time you make a bond, now if you can see, this is a cyclic structure already formed here, right? Close from all the end. How many atoms you have in there? 1 2 3 4 5 6. Out of the 6, 1 is oxygen, 5 is carbon. The best structure is hexagonal. This is your hexagonal, right? And then we're gonna put the groups on each carbon. So this is your carbon number 1 2 3 4 and 5, right? So this is 1, this is 2, this is 3, this is 4, this is 5, this is 6. Let's do on carbon number 1. How many groups is attached? On carbon number 1 you have OH and hydrogen. So this OH you can put on the top, hydrogen on the bottom, or you can flip, vice versa. On the second carbon, OH on the right hand side below the ring, hydrogen on the top. On the third, hydrogen on the right below the ring, OH on the top. On the fourth, OH on the right below the ring, hydrogen on the top. On the fifth carbon, one of the bond is attached with the oxygen, which is this. Another bond attached with the hydrogen and another bond with CS2OH. So remember this thing, this CS2OH always goes on the top and the hydrogen on the bottom. So what's the name of this? So if you remember, this is your anomeric carbon. Anomeric carbon, and OH is on the top, then this is beta, and this glucose comes from where? D glucose. So beta D glucose, right? So this is one of the isomer, but this OH could have been on the bottom, H could have been on the top. Then if it was the case, then it will be alpha D glucose. So just remember this thing, right? This OH can be go down, this H can be go on up. So that's why you have alpha and beta isomers. So this is how you normally draw from a Fisher projection to a Haworth projection. Haworth, right? The cyclic structure. Let's do one more example with the fructose, the second kind of monosaccharide. So that's your fructose, CS2OH. Then you have carbon, C double bond O. Then you have H, OH. Then you have OH, H. Then you have OH, H. And then at the last you have CS2OH. And this is your D-fructose. So how you're going to convert this D-fructose in a Fisher projection into a Haworth projection, which is a cyclic ring structure. Again, the same principle. Take this is your last chiral center carbon with an OH. Take this hydrogen, put on this oxygen, which is the most oxidized carbon, right? You cannot put on the first carbon because you always do on the carbonyl group, always. So now I'm gonna remove this hydrogen from here because it's already on the oxygen, and then I'm going to remove one of the bond, right? Because oxygen already got it. Now this oxygen is going to bind with this oxygen, with this carbon, and now you're gonna make a cyclic ring, including this carbon, right? This is a cyclic, and it start from carbon number 2, 3, 4, 5, 6. So if you have 5, then 5 is something like this with a pentagon, right? And then let's try to put, so this oxygen bound with second carbon. So this is your second carbon. This is 3, this is 4, this is 5. So let's put on the second carbon you have CS2OH attached, and you have OH. On the third, H is on the bottom, OH on the top. On the fourth, you have OH on the bottom, H on the top. On the fifth, you have oxygen, which is already over here. then hydrogen and CH2OH, and I told you CH2OH will always be on the top and hydrogen. So this is your which isomer. In this case OH is on the bottom, then this is alpha came from D fructose, alpha D fructose, right? And then like I said on the previous slide, this OH could have been on the top and CS2OH could have been on the bottom, right? Something like this. And everything remained the same, right? So this special case is beta D fructose, because of the OH is on the top of the anomeric carbon. This is your anomeric carbon, right? We already talked about the anomeric carbon. So yeah, so make sure you practice to draw a couple of structure from fructose to, or from the feature projection to any structure. So if you have the feature projection for any carbohydrate monosaccharide, you can draw the ring-like structure, the Howard projection. Thus, if you apply the same concept or same steps that we did together right now. Now let's try to do some problems and then with the concept that we just learned. So let's start with this problem. Answer the question on the for the two rings formed on D galactose. A. Identify alpha D galactose and beta D galactose. Are they enantiomer or diastereomers? So if it says alpha and beta, I need to go to the anomeric carbon. So this OH is on the bottom, this is alpha, OH on the top, this is beta. Are they enantiomer? They are diastereomers. Right. Number each carbon atom in the ring. Which carbon atom is the anomeric carbon? We already talked about. This is your anomeric carbon. This is your anomeric carbon. How many carbon atom you have in there? 1, 2, 3, 4, 5. You have 5 carbon atom in the ring. Are these furanose or pyranose? This is a pyranose because you have 6-member ring. From the Howard projection, how can you tell these are the D sugar? From the feature projection, how can you tell is a D sugar? So this is a D sugar because it's come from the D galactose. Right. So this is how you normally solve the problem, and make sure you practice this kind of problem too. Right. Now let's talk about the another subtopic, which is the structure of the complex carbohydrate. So we have different kind of complex carbohydrate. It can be disaccharide, oligosaccharide, or polysaccharide because it's made up of 2 or more monosaccharide, right? And how, if they are made up of 2 or more saccharide, they're gonna combine through a bond, because if you have to combine 2 molecules of glucose together, there has to be some kind of bond, and that kind of bond we call as a glycosidic bond. So this is the bond that combine 2 or multiple single sugar molecule together, and we're gonna see how the individual monosaccharide combine together through this glycosidic bond to make this oligo or polysaccharide molecule. So let's go to the next slide and try to learn the first kind of complex carbohydrate, which is a disaccharide. So we're gonna talk about the disaccharide carbohydrate that undergo hydrolysis to form 2 monosaccharide. We already learned about this definition on the couple of first slide. So we have 3 different kind of disaccharide. One is called maltose, second one is called sucrose, and the third one we call as lactose. If you do a hydrolysis to break a bond, you're gonna get glucose and galactose. So make sure you remember these different kind of disaccharide. Maltose, whenever you do hydrolysis, you will get 2 molecules of same glucose. Sucrose give you glucose plus fructose, and then lactose, glucose plus galactose. So now we're gonna talk about what information does glycosidic linkage gives us. So first, it normally gives where on the rings are the two monosaccharides joined. It will give you the number. Second, it will tells you the anomeric center, whether it's alpha or beta, right? For example, a lactose, one of the disaccharide, which is also called as a milk soda, has a glycosidic linkage of beta 1,4 linkage. So we're going to learn about what does this mean, beta 1,4, right? Let's go to the next slide and try to learn what is this beta 1,4 linkage, glycosidic linkage mean. So now let's talk about the beta linkage in galactose, which is made up of a galactose and glucose. So you have two monosaccharide, D-galactose and D-glucose, and on the first carbon, before it made a glycosidic linkage, this kind of linkage, it had like OH group over here, and the fourth carbon on the glucose has the OH group. And once they are ready to make a linkage between the two molecules, then the H and OH from two different monosaccharide from carbon number 1 and 4 combine together to make a water, and then the oxygen come in between to make a glycosidic bond, right? So you're gonna make something like this with an oxygen at the center, and then connecting carbon number 1 and carbon number 4, right? And this is your glycosidic linkage. So if the name of the glycosidic linkage is 1 and 4, that means the carbon number 1 and carbon number 4 from different monosaccharide combine together to form this glycosidic linkage. And what is this beta means? Beta means the carbon number 1, the OH is present on the top. So the anomeric carbon has an OH on the top. That means it's a beta isomer. So the beta isomer of the galactose combining with an OH from the top to make water as a product and combine as a glycosidic linkage. So that's why it's a beta isomer. So beta 1,4 glycosidic linkage. So make sure you remember this kind of linkage in a lactose. And if you can see the second anomeric carbon from on the second monosaccharide Howard projection, which is a glucose, this is free, right? It's not bound to any of the other monosaccharide, right? So if you dissolve this lactose molecule in a water, so this glycosidic linkage won't break down, but this anomeric carbon can open one more time, and then this thing convert into a aldehyde again, right? So this is an equilibrium with the Fischer projection, so it can convert into aldehyde again into a Haworth projection. So they are in equilibrium, right? And then once it convert into aldehyde, it can do some oxidation or reduction reaction. So that's why we call lactose as a reducing sugar, because it has an aldehyde group once it's opened back into a Fischer projection, right? Now let's talk about the bond in sucrose, which is a disaccharide. Sucrose is made up of glucose plus fructose, right? So this is your fructose, this is your glucose, right? And if you can see which kind of bonding you have in here, you have alpha 1 beta 2 glycosidic linkage, right? Or glycosidic bond. Why? Because if you see on the fructose, this is your anomeric carbon, and in here the OH was over here before it converted into a glycosidic bond. And in glucose, in anomeric carbon, this is your anomeric carbon. In fructose, this is your anomeric carbon, right? The OH was over here. When it combined to make water as a byproduct, you left with this bond, carbon, oxygen, carbon, right? So I'm going to remove this thing, and then you end up with this glycosidic bond. So over here, if you can see, both the anomeric carbon on each monosaccharide, right, the top one and the bottom one, both are involved in the bonding of the glycosidic bond. So that's why it's alpha 1 means on the first monosaccharide on the carbon number 1, the configuration was alpha because OH was on the bottom, and then on the fructose on anomeric carbon, OH was on the top, that why is a beta. So that's why the bond is alpha 1 beta 2 glycosidic bond, right? And what's the second important thing over here? In both these structure, the anomeric carbon are making a glycosidic bond. So that means in both the monosaccharide Howard projection, there is no anomeric carbon which is freely available. If they are not freely available, they cannot convert to an aldehyde or ketone. If they cannot convert into aldehyde or ketone in a Fischer projection, when you dissolve in there, then that means they cannot reduce or oxidize anything. So that means sucrose is a non-reducing sugar. So just remember this thing, because there is no anomeric carbon that is freely available, because only the anomeric carbon are in equilibrium with the open chain. But in this case, these are involved in the bond formation. That's why sucrose is a non-reducing sugar. So remember this thing. And which kind of bond you have? Alpha 1 beta 2 glycosidic bond. So these are some of the common disaccharide. We already talked about the lactose, sucrose. Then we have maltose. In maltose, which kind of bond we have? Alpha 1 4, because the first anomeric carbon bind with the fourth carbon of the second monosaccharide through a glycosidic bond, right? And then this anomeric carbon is free. It can convert into an aldehyde whenever you dissolve in water, and aldehyde can reduce another stuff or oxidize another stuff. So that's why this is also a reducing sugar, maltose, but sucrose not. So make sure from this table, make sure what are the individual monosaccharide making that disaccharide, what are the different kind of bond we have, and make sure you know the structure, right? So this is a summary of all the disaccharide. Now let's talk about the last kind of carbohydrate, which is a polysaccharide. As we already learned before, polysaccharide are biological polymer, large molecule composed of repeating structural unit called monomer, right? Or made up of different monosaccharide, right? Most polysaccharide are composed of D-glucose but differ in the type of glycosidic linkage. So how many different types of polysaccharide are different from each other is a type of glycosidic linkage, how they are combining with the other glucose molecule. And there's a couple of examples: starch, glycogen, and cellulose. These are the three polysaccharide example. Let's talk about these things on the next slide. So now let's talk about the starch. Starch has 250 to 400 glycosidic linkage, and its starch is made up of two components. One is called amylose, second one is called amylopectin, and amylopectin is present in high proportion compared to the amylose. And when you digest, it is hydrolyzed into a D-glucose, because starch is made up of a multiple unit of glucose attached through a glycosidic bond, right? And if you have an excess D-glucose in your body, it is going to convert into a glycogen as a stored glucose, right? And is stored in the liver or muscle cells. Right? This is how body normally reacts if you have high glucose level. Now let's talk about the two components of the starch, amylose and amylopectin. Amylose unbranched chain, all are alpha 1,4 glycosidic linkage, means every glucose molecule that we have, which kind of bond you have in between them, you have alpha 1,4. Right? Second is amylopectin. Amylopectin also has alpha 1,4. Right? If you have multiple glucose molecule or a monomer, but along with the alpha 1,4, it also has alpha 1,6. You will have some kind of branching, right? So this is a branch polymer, right? Whereas amylose is a single line structure, right? A linear structure. And then there is a similarity between glycogen. What is glycogen? Is a stored form of the glucose. It is similar to the amylopectin but larger and more highly branched. So how the glycogen is different from the amylopectin, which is a starch, is that they are larger, they have more monosaccharide unit combined together, and they are more branched, right? That's the difference. The branching is high in glycogen compared to the amylopectin, but beside that everything is same. Then we have the last, which is a cellulose, polysaccharide. Cellulose, as we know, main component of the wood. You can find this thing in the wood, paper, cotton, and it's made up of a monosaccharide of glucose. And cellulose, as we already discussed, made up of a D-glucose, and it has a glycosidic linkage beta 1,4. So that means if you have a glucose molecules, these are the glucose monosaccharide, and which kind of bonding you have in between them, you have beta 1,4. And what's the purpose of cellulose in the plant? It gives the structural strength to the plant, right? And it has some structured role, and normally hydrolyzed by beta glycosidase. So the enzyme that is going to break this bond is beta glycosidase, which human doesn't have this beta glycosidase. So if the human doesn't have this enzyme, so we cannot eat wood, paper or cotton, because we don't have an enzyme to break down this bond to hydrolyze this polysaccharide. But certain insect and bacteria do have this beta glycosidase, so that's why they can eat wood. So make sure you know the difference, right? So now let's try to do this problem based on the concept that we just did. So just fill it out the blanks that is given over here. It says for each of the statement below indicate the polysaccharide to which the statement apply. Dash is the storage form of glucose in mammal cell. So it's gonna be third. Glycogen is in the form that we normally stored glucose in livers. Then second one is cannot be digested by human. This is your cellulose, which is 4. Then we have the third one is also known as starch. So amylose and amylopectin, both is a form of a starch. So you can say 1 and 2. Dash is produced by plant through photosynthesis. So you can say amylose, amylopectin, and then you can say cellulose, right? So you can say 1, 2 and 4. Then the third one, second last was has a helical overall shape. That's your amylose. And then the last one is hydrolyzed by alpha glycosidase. So everything is gonna hydrolyze by alpha because all of them has alpha glycosidic linkage, but cellulose has beta glycosidic linkage, so this cannot be hydrolyzed. So this is 1, 2, 3. So this is how you can match or fill up the blank spaces from all the four options that you have it in here. Now let's go to our last topic from this PowerPoint, which is the role of the oligosaccharide as cell marker. We already talked about what are oligosaccharide. Is a combination of few monosaccharide unit together that make up the oligosaccharide, and it can be used as a cell marker. So what is cell marker? Cell marker has a unique carbohydrate chain on the surface of our cell. So we know our cell is made up of or has a covering called cell membrane, and on the cell membrane you have this oligosaccharide present along with the protein and other biomolecule. We're gonna see on the next slide that how this oligosaccharide plays on the surface of the cell membrane, and it helps to allow immune cells to distinguish ourselves from the cell of other organism. So this also act as a cell marker. So immune cells know that this kind of oligosaccharide is present in our cell but not on the other cell, right? And then most of the oligosaccharide bonded to the protein or lipid in a cell membrane, and cause of rejection post organ transplant, because if somebody has an organ transplant already done, they have the different oligosaccharide present on the cell membrane compared to the cell that person already has. So if an immune cell goes to the organ that has been transplanted, they have a different oligosaccharide, and then the immune cell will think that this is a different cell and might attack those cells. So that's why the person who got the organ transplant always need to take immunosuppressant, right? So let's go to the next slide and see the structure of the oligosaccharide, which act as a cell marker. So this is the structure of the cell membrane. So you will see these are your oligosaccharide, which act as a cell marker, right? Then you have a protein, you have your lipid membrane over here, right? So this is act as a cell marker to differentiate own cell compared to the other organism cell. So let's take an example where we can use oligosaccharide as a marker. We can take an example of a blood cells, right? Blood groups, you can say type A, B, AB and O blood cell, and they are all different because of the different oligosaccharide present on the RBC, red blood cell. So type A blood, types O blood cell has trisaccharide, means 3 glucose molecule or through 3 saccharide monosaccharide molecule over there. Type A blood has tetrasaccharide marker with same 3 as you found in O, but the fourth one is different. That is not found in O and type B. Then type B has tetrasaccharide one more time with the same 3 as O plus a monosaccharide not found in O and A. So the fourth one is the differentiation between A and B. Then we have type AB contain both type A and type B marker, right? So depending on which kind of oligosaccharide you have on the surface of the cell membrane of RBC, we have a different kind of blood groups. Let's go to the next slide and then see the pictorial representation of these groups. So we have these blood types and oligosaccharide marker. Let's say these are the O, A, B and AB groups, and then this is a RBC cell membrane. You have this is a trisaccharide, you have one glucose, one saccharide, second carbohydrate, third carbohydrate. Then A, we have the same, but the fourth one is different. For B, again the 3 are same, the fourth one is different than B, right? This is different. Then we have AB, we can have these 3 same, and the fourth one, then B, 1, 2, 3, and the same D-galactose, right? So they have both of them. So this is how the oligosaccharide arrange on the surface of the RBC. And if we can see over here on the last column, we can say O is a universal donor. Why? Because these 3 trisaccharide that make up the O blood type present in every other group, A, B and AB, if you can see the common, right? Then B blood group can donate to the B and O because B blood group has the same composition of the oligosaccharide as B and O. Then AB, you can say, is a universal recipient because it has everything that is present in every other blood group, O, single A, single B. So it's a universal acceptor of the blood. So this is how you can differentiate between the different blood group side. This is how you can say which one is a universal donor and why, and which one is a universal acceptor or recipient and why is the case. So we already talked about this thing on the previous slide too. Type O patient is a universal donor because it can donate to recipient of any blood group, right? And type AB is your universal recipient, and we already talked about in the previous slide why is the case, right? So let's go to the last slide and then try to do one problem and then try to understand the concept that we just learned. So let's try to do this problem. Can people with a type B blood accept a blood donation from someone with O? Can someone with type O blood accept a blood donation from someone with type B? Explain. So the first question is yes, type O donor are universal donor. That's why it can donate to any of the other blood group, right? Because there is no danger of immune response. Whereas in the question 2, can people with type O blood accept from the type B? It says no, because O blood can accept blood only from O. Type B blood would cause an immune response in the type O individual because it has one extra carbohydrate molecule in type B which was not present in O. So that's why the cell or the immune system might think that this blood is a foreign blood or foreign cell. So this might lead to a clotting because of the immune response. So this is how you can tell the answer depending on which type of patient will get which kind of blood from different blood groups patient. So yeah, so this is the end of the topic or end of the chapter. Make sure you read it from the textbook too, and then see the video that you are seeing right now, and yeah, and then I'll see you in the next video. Bye.